Similarly, cooperative hunting in lions or alarm calling in primates can be seen as increasing the survival of the group, which may outcompete less cooperative groups. In human evolution, group selection has been invoked to explain the emergence of traits such as cooperation, morality, and social norms. Human groups that were more cooperative likely outcompeted others in warfare or resource acquisition. Cultural group selection is a modern extension, where cultural traits that favor group cohesion spread because of group-level advantages. It is important to differentiate naive group selection, now largely rejected, from multi-level selection, which is mathematically robust and accepted in modern evolutionary biology. Multi-level selection is that selection can act simultaneously at multiple hierarchical levels: genes, individuals, groups, even species, depending on the ecological and genetic structure of populations. In summary, while individual selection remains the primary force in evolution, group selection or multi-level selection is accepted as a valid evolutionary framework when group-level differences in survival and reproduction significantly influence gene frequencies. For the CSIR-NET exam, one should focus on the historical debate, mathematical principles, examples from natural populations, and the integration of group selection into the broader modern synthesis of evolutionary theory. Previous year questions, CSIR-NET Life Sciences, December 2015, Part B. Group selection refers to natural selection acting at the level of individuals. For natural selection acting at the level of individuals. B. Groups of individuals. C. Genes. D. Populations without group structure. Answer, B. Explanation. Group selection acts on traits that benefit the group as a whole. June 2017, Part C. In a population divided into multiple groups, altruists benefit the group but have lower individual fitness than selfish individuals. What mechanism can maintain altruism in the population? A. Kin selection. B. Intergroup selection. C. Group selection. D. Frequency-dependent selection. Answer, C. Explanation. When altruistic groups outcompete selfish ones, group selection can maintain altruism. Mock Test 2023, CSIR website. Which statement best describes multi-level selection theory? A. Selection acts only at the level of genes. B. Selection acts only at the level of individuals. C. Selection can act simultaneously at the level of individuals and groups. D. Selection at the group level eliminates individual. Selection at the group level eliminates individual levels. Select answer, C. Explanation. Multi-level selection acknowledges the simultaneous action of selection at different biological levels. Let me know the next topic you want, whether it's kin selection, reciprocal altruism, cultural evolution, or any. Let me know the next topic you want, whether it's kin selection, reciprocal altruism, cultural evolution, or anything else from evolution or behavior. I'll give you a full, paragraph-based lecture designed solely to help you crack CSIR-NET JRF Life Sciences. You said, kin selection, ChatGPT said. Here's your CSIR-NET JRF Life Sciences lecture on kin selection, written at JRF level depth, paragraph style, no tables, with maximum word limit coverage, based on previous year CSIR questions, and tailored to be your only coaching material for the exam. Kin selection, CSIR-NET JRF Life Sciences full theory lecture. Kin selection is a form of natural selection where individuals increase their genetic fitness not only by producing their own offspring but also by helping relatives reproduce, because relatives share common genes. One idea is that genes promoting altruistic behaviors can spread in a population if those behaviors increase the reproductive success of kin who carry the same genes. The concept formally introduced by W. D. Hamilton in the 1960s, and it revolutionized our understanding of social behavior in animals. At the heart of kin selection is Hamilton's rule, which provides the condition under which an altruistic trait will evolve. It is mathematically expressed as RB and GT, C, where R is the coefficient of relatedness between the actor and the recipient, B is the reproductive benefit gained by the recipient of the altruism, and C is the reproductive cost to the actor. If the benefit to the relative weighted by the degree of genetic relatedness outweighs the cost to the altruist, then the behavior can be favored by natural selection. The coefficient of relatedness R represents the probability that two individuals share a gene by common descent. For example, R equals 0.5 for siblings or parent offspring, R equals 0.25 for half-siblings or grandparent-grandchild, and R equals 0.125 for first cousins. Kin selection explains the evolution of altruism among relatives, where direct fitness is sacrificed. The coefficient of relatedness R represents the probability that two individuals share a gene by common descent. For example, R equals 0.5 for siblings or parent offspring, R equals 0.25 for half-siblings or grandparent-grandchild, and R equals 0.125 for first cousins. The coefficient of relatedness R represents the probability that two individuals share a gene by common descent. For example, R equals 0.5 for siblings or parent offspring, R equals 0.25 for half-siblings or grandparent-grandchild, and R equals 0.125 for first cousins. Kin selection explains the evolution of altruism among relatives, where direct fitness is sacrificed to increase indirect fitness. This combined measure of direct and indirect fitness is termed inclusive fitness, and natural selection acting to maximize this inclusive fitness is what underpins kin selection. The concept is particularly useful in explaining behaviors that appear maladaptive at the individual level but are adaptive when considered in the context of shared genes. A striking example is found in eusocial insects such as bees, ants, and wasps. In the species, sterile worker females forego reproduction to assist the queen in rearing more siblings. In Hymenoptera, sex determination is haplodiploid, where females are diploid and males are haploid. This results in sisters sharing 75% of their genes, which means a female worker can pass on more of her genes by helping her mother, the queen, raise sisters than by reproducing herself. This high relatedness is a key factor in the evolution of complex eusocial and caste systems in these insects. Kin selection also operates in vertebrates. In cooperative breeding birds like the Florida scrub jay, offspring often stay with their parents and help raise younger siblings instead of breeding on their own. This reproduction is adaptive because the helpers increase the survival of close kin and may later inherit territory or breeding positions. In mammals like meerkats, cooperative rearing of young and sentinel behavior are often performed by individuals helping close kin, thereby increasing inclusive fitness. It's important to understand that kin selection does not imply conscious awareness of genetic relatedness. Instead, selection favors behavioral mechanisms such as imprinting, spatial proximity, or chemical cues that bias helping behavior toward kin. In humans, kin selection may underlie strong family bonds and caregiving behavior among close relatives, although culture and reciprocity add complexity. Kin selection differs from group selection in that it focuses on gene propagation via relatives rather than group-level traits. It also differs from reciprocal altruism, where individuals help others with the expectation of future help, typically among unrelated individuals. Kin selection can act independently or alongside these other mechanisms in the evolution of social behavior. In modern evolutionary biology, kin selection remains one of the most powerful and widely accepted explanations for altruism, cooperation, and social structure, especially in species with stable family units. Understanding kin selection is crucial for interpreting animal behavior patterns, mating systems, life history strategies, and the evolution of complex societies. Previous year CSIR-NET questions on kin selection, December 2013, Part B. The concept of inclusive fitness is used to explain a selection at the species level. B. The evolution of altruistic behavior among relatives. C. Mutation pressure as a driving force of evolution. D. Adaptive radiation in island habitats. Answer B. Explanation. Inclusive fitness includes both direct and indirect fitness, explaining altruism among kin. June 2015, Part C. A sterile female worker bee helps her mother, the queen, produce more daughters instead of reproducing herself. According to kin selection theory, which of the following best explains this behavior? A kin selection promotes behaviors that reduce inclusive fitness. C. Worker bees are more related to their sisters than their own offspring. C. Worker bees gain direct fitness through helping. D. Selection acts only on groups in eusocial insects. Answer B. Explanation. In haplodiploid systems, sisters share 75% genes, more than mother-offspring relatedness. December 2022, Part C. According to Hamilton's rule, an altruistic trait is expected to evolve if AR and GT, B, C. BC and GT, RBC, RB and GT, CD, RB and LT, C. Answer, C. Explanation. Hamilton's rule. Altruism evolves if benefit to kin, RB, exceeds cost, C. Let me know the next topic you want, such as reciprocal altruism, evolution of social behavior, biological clocks, or sensory ecology, and I'll prepare the same ultra-detailed, JRF level lecture for you. You said. Reciprocal altruism, ChatGPT said. Here is your CSIR-NET JRF Life Sciences lecture on reciprocal altruism, written in maximum detail, no table, paragraph-based format, tailored to the JRF level, including 2 to 3 previous year questions with answers in years. This is crafted to be your only study material for cracking the CSIR-NET Life Sciences exam. Reciprocal altruism, CSIR-NET JRF Life Sciences Full Theory Lecture. Reciprocal altruism is a form of cooperation in which an organism acts in a manner that temporarily reduces its own fitness while increasing another's fitness, with the expectation that the favor will be returned in the future. This concept was introduced by Robert Trivers in 1971 and extended the theory of altruism beyond genetically related individuals, as explained by kin selection. It provides an evolutionary framework to understand altruistic behavior between non-relatives, which cannot be explained by shared genetic material alone. The key conditions that favor the evolution of reciprocal altruism include repeated interactions between the same individuals, recognition and memory of past interactions to detect cheaters, a benefit to the recipient that is greater than the cost to the actor, a mechanism of retaliation if cooperation is not reciprocated. In reciprocal altruism, the cost incurred by the altruist is offset by the future benefit of receiving help, assuming a system of mutual cooperation is maintained over time. This concept can be observed in various animal behaviors where the actor's investment is eventually compensated by a returned act of assistance, leading to long-term mutual benefit. One of the best-known examples comes from vampire bats, which share blood meals with roost mates that have failed to feed. The sharing bat incurs a short-term cost by regurgitating blood, but this act is typically directed towards individuals who have previously shared with them. Studies show that bats remember past donors and are more likely to help those that have helped them before, even among non-kin, a classic case of reciprocal altruism. Another well-studied system involves primates. In species such as chimpanzees and baboons, individuals engage in grooming, which not only improves hygiene but strengthens social bonds. Grooming is often reciprocated. An individual that grooms another is likely to receive grooming in return. Moreover, grooming can be exchanged for other favors such as support in fights, mating opportunities, or food sharing. These social exchanges, though not simultaneous, create a network of delayed but mutual benefits that enhance the overall fitness of individuals in the group. From an evolutionary standpoint, reciprocal altruism can be vulnerable to cheating, where an individual accepts the benefit but fails to reciprocate. However, natural selection favors strategies to minimize cheating, such as punishment, exclusion from future cooperation, or partner switching. This leads to the evolution of trust, fairness, and social rules, especially in cognitively advanced species. In humans, reciprocal altruism is deeply embedded in social behavior, morality, and cooperation, and is often formalized in cultural practices. We engage in acts like lending, gift-giving, and cooperative labor, expecting future reciprocation. Complex societies have developed norms and legal systems to enforce and encourage reciprocal behavior, making it a cornerstone of social evolution. Reciprocal altruism is not driven by shared genes but by shared interest in future interactions. It has been modeled extensively using game theory, particularly the prisoner's dilemma, where the best strategy for long-term interaction is tit for tat, start with cooperation and then replicate the partner's previous move. These models show that cooperation can evolve and be stable, even in populations of unrelated individuals, under repeated interaction scenarios. In summary, reciprocal altruism provides an evolutionary explanation for non-kin cooperation, especially in social animals. It relies on memory, recognition, and long-term interactions, and is a key element in the evolution of complex social behavior. For CSIR-NET, understanding reciprocal altruism in contrast with kin selection, group selection, and selfish behavior is essential. Previous year questions, CSIR Net Life Sciences, June 2013, Part B. Reciprocal altruism differs from kin selection in that it involves cooperation among unrelated individuals. B. It leads to inclusive fitness. C. It occurs only in eusocial insects. D. It is based on genetic relatedness only. Answer A. Explanation. Reciprocal altruism occurs between non-kin based on repeated interactions. December 2016, Part C. Which of the following conditions is necessary for the evolution of reciprocal altruism? A one-time interaction between individuals. B. High mutation rates in population. C. Long-term association and memory of past behavior. D. High relatedness among group members. Answer C. Explanation. Memory and repeated interactions are essential for reciprocal altruism. Mock test CSIR, 2022. A vampire bat shares food with another that is not related, but has shared food before. This is an example of the kin selection. B. Reciprocal altruism. C. Group selection. D. Altruism based on inclusive fitness. Answer B. Explanation. This is cooperation among non-kin, expected to be reciprocated in future. Let me know if you want the next lecture on biological clocks, sensory ecology, evolution of social behavior, or any other CSIR topic. I'll give you a full JRF level, one stop note again. You said. Neural basis of learning, ChatGPT said. Here is your CSIR Net JRF Life Sciences lecture on neural basis of learning, prepared at JRF level, maximum word limit, paragraph based, with no tables, and includes 2 to 3 previous year CSIR questions with answers in years. This is designed to serve as your complete and only coaching material for this topic. Neural basis of learning, CSIR Net JRF Life Sciences full theory lecture. Learning is the process through which experience leads to a relatively permanent change in behavior or the potential for behavior. The neural basis of learning refers to the physiological and structural changes that occur in the nervous system, particularly the brain, during the acquisition and storage of new information. At the core of learning lie synaptic modifications, neurochemical signaling, and changes in neuronal circuitry. One of the most fundamental principles underlying learning is synaptic plasticity, the ability of synapses to strengthen or weaken over time in response to increases or decreases in their activity. This plasticity enables the nervous system to encode experiences, form memories, and modify behavior accordingly. Two primary forms of synaptic plasticity that are critical to learning are long-term potentiation, LTP, and long-term depression, limited. Long-term potentiation, LTP, is a long-lasting enhancement in signal transmission between two neurons that results from their simultaneous stimulation. It is considered a key cellular mechanism underlying learning and memory, especially in the hippocampus, which is involved in spatial and declarative memory. LTP typically occurs at glutamatergic synapses and is mediated by NMDA and methyl D-aspartate receptors, which are both voltage-gated and ligand-gated. When presynaptic glutamate release coincides with postsynaptic depolarization, NMDA receptors allow calcium influx, initiating intracellular signaling cascades that strengthen synaptic connections, either by increasing receptor density or altering gene expression. In contrast, long-term depression, limited, involves a long-lasting decrease in synaptic strength. It also occurs at glutamatergic synapses but often involves a low-frequency stimulation that results in a modest but sustained calcium influx. This activates different pathways, e.g., phosphatases instead of kinases, leading to synaptic weakening. The balance between LTP and limited is essential for synaptic scaling, memory formation, and forgetting, thereby refining learned responses. Another key player in the neural basis of learning is Hebbian plasticity, often summarized by the phrase, cells that fire together, wire together. According to this principle, if a presynaptic neuron repeatedly activates a postsynaptic neuron, the synaptic connection between them strengthens. Hebbian learning provides the framework for associative learning, including classical conditioning and operant conditioning. In non-associative learning such as habituation, decreased response to a repeated stimulus, and sensitization, increased response after a strong or noxious stimulus, specific changes at the neuronal level have also been documented. These forms of learning have been extensively studied in simple systems like aplysia, a C-slug used in neurobiology. In aplysia, habituation involves reduced neurotransmitter release at synapses, while sensitization involves enhanced neurotransmitter release due to modulatory interneurons and the involvement of second messengers like cyclic AMP. Neurotransmitters and neuromodulators are critical in learning mechanisms. For instance, dopamine is essential in reward-based learning and reinforcement, particularly via its action in the mesolimbic pathway, which includes the ventral tegmental area (VTA) and the nucleus accumbens. The role of dopamine in learning is not just about pleasure but also about prediction error, a mismatch between expected and actual outcomes, which updates future behavior. Brains involved in learning include the hippocampus, declarative and spatial memory; the amygdala, emotional learning, fear conditioning; the cerebellum, motor learning and classical conditioning; the prefrontal cortex, working memory, decision-making, and extinction of learned behaviors; and the basal ganglia, procedural learning and the basal ganglia, procedural learning and habit formation. Other organisms supporting learning include gene expression and protein synthesis, particularly during the consolidation phase. Transcription factors like CREB, GAMP response element binding protein, are crucial for converting short-term plastic changes into long-term memory. Blocking protein synthesis often prevents long-term memory formation, demonstrating the link between molecular processes and behavioral learning. Learning also modifies the structural architecture of the brain. Studies using imaging and microscopy have revealed synaptogenesis, dendritic branching, and even adult neurogenesis in areas like the hippocampus, all associated with learning. In conclusion, the neural basis of learning involves a dynamic interplay of electrical activity, molecular signaling, synaptic remodeling, and structural changes in the brain. Understanding this complexity is central to the study of behavioral neuroscience, neurodevelopment, and neurodegenerative disorders that impair learning and memory, such as Alzheimer's disease. Previous year CSIR Net questions on neural basis of learning, June 2014, Part B. Long-term potentiation (LTP) in the hippocampus is associated with a decrease in synaptic strength. B. Increase in postsynaptic receptor density. C. Blockade of calcium influx. D. Decreased neurotransmitter release. Answer B. Action. LTP involves insertion of AMPA receptors into the postsynaptic membrane, increasing synaptic strength. December 2015, Part C. In an experiment on aplesia, repeated gentle stimulation of the siphon results in reduced gill withdrawal. This is an example of the classical conditioning. B. Habituation. C. Sensitization. D. Extinction. Answer B. Explanation. Aplesia shows reduced response to repeated, benign stimulus, a hallmark of habituation. June 2019, Part B. Component of synaptic plasticity underlying learning is a death of neurons in the hippocampus. B. Static synaptic strength over time. C. Activity-dependent changes in synaptic strength. D. Inhibition of neurotransmitter synthesis. Answer C. Explanation. Learning requires activity-dependent modifications of synapses, such as LTP and limited. Let me know if you'd like the next topic: biological clocks, molecular basis of memory, evolution of nervous system, or any other CSIR life sciences topic. I'll prepare it with the same quality. You said, memory, ChatGPT said. Here is your CSIR Net JRF Life Sciences lecture on memory, written at the highest possible quality and depth, suitable for JRF level preparation. It is crafted entirely in paragraph form, with no tables, includes 2 to 3 previous year CSIR Net questions with answers in year mentioned, and is designed to serve as your only study material for cracking the exam. Complete theory for CSIR Net JRF Life Sciences. Memory is the fundamental process by which organisms encode, store, and retrieve information. It is a vital function of the nervous system that allows an individual to adapt, learn from past experiences, and make decisions. At its core, memory is a consequence of neuroplasticity, whereby synapses, neuronal networks, and entire brain structures undergo biochemical, electrical, and morphological changes in response to experience. Memory can be categorized into various types based on time scale, consciousness, and neural circuits involved, including sensory memory, short-term memory, working memory, and long-term memory, which can be further divided into explicit declarative and implicit non-declarative forms. Sensory memory is the most immediate form of memory, lasting less than a second. It thoroughly retains sensory impressions, like visual, iconic, and auditory, echoic information, and serves as the initial filter before the information is processed further. Sensory memory depends largely on activity in the primary sensory cortices. Short-term memory, STM, retains limited information for a brief duration, usually seconds to minutes. The classic example is remembering a phone number long enough to dial it. Working memory is a refined component of STM that not only stores but also manipulates information in real time, such as solving a math problem mentally. Memory is highly dependent on the prefrontal cortex, where executive control, attention regulation, and integration of multiple cognitive processes occur. Long-term memory, LTM, refers to the enduring storage of information for days, years, or a lifetime. It involves 2 major subtypes: explicit memory, which requires conscious recall, and implicit memory, which operates unconsciously. Explicit memory includes episodic memory, personal experiences, and semantic memory, facts and knowledge. These types are primarily associated with the hippocampus, medial temporal lobe, and neocortex. The hippocampus is critical for the consolidation of short-term episodic memories into long-term storage. Lesions in this area, as seen in the famous case of Henry Molaison, HM, result in profound anterograde amnesia, loss of the ability to form new long-term memories. Implicit memory includes skills and habits, procedural memory, classical conditioning, and priming. It relies on brain areas like the basal ganglia, cerebellum, and amygdala. Procedural memory enables learning of motor sequences, e.g., riding a bicycle, and does not require conscious recall. At the cellular level, memory formation involves changes in synaptic strength via processes like long-term potentiation, LTP, and long-term depression, LTD, particularly in the hippocampus. LTP, triggered by high-frequency stimulation, leads to increased calcium influx through NMDA receptors, activation of CA-McKee and PKC, and subsequent insertion of AMPA receptors into the postsynaptic membrane, resulting in stronger synaptic responses. Over time, LTP induces gene expression changes in protein synthesis necessary for stabilizing synaptic modifications, a process called memory consolidation. Transcription factors like CREB, GAMP response element binding protein, are essential for initiating these gene expression cascades. It's immediately after learning impairs long-term memory but not short-term memory, reinforcing the molecular distinction between memory stages. In addition to the hippocampus, sleep plays a major role in memory consolidation, especially for declarative memories. During slow-wave sleep, hippocampus-dependent memories are repeatedly reactivated and redistributed to cortical sites for long-term storage. It's a 2-stage model of memory, where hippocampal networks initially encode information, which is later transferred to the neocortex for permanent retention. Forgetting, often misunderstood as memory failure, can be an active process, necessary to prevent cognitive overload and optimize decision-making. Forgetting involves mechanisms like synaptic pruning, limited, and inhibitory regulation of memory retrieval circuits. Prefrontal cortex modulates the recall and suppression of memories, balancing memory retention with adaptability. Neurodegenerative diseases, such as Alzheimer's disease, are characterized by severe memory impairments. Alzheimer's typically begins with loss of episodic memory, progressing to semantic and procedural deficits. Pathological hallmarks include amyloid beta plaques, neurofibrillary tangles, and hippocampal atrophy, all of which disrupt the neural circuits essential for memory. In sum, memory is a multi-layered process involving encoding, storage, and retrieval of information through complex and interdependent neural, cellular, and molecular mechanisms. From synaptic plasticity to large-scale neural networks, memory illustrates how behavior, experience, and biology are intimately connected. Understanding its neural basis is not only central to neuroscience but also critical for addressing cognitive disorders, optimizing learning strategies, and developing therapeutic interventions. Previous year CSIR Net questions on memory, CSIR Net June 2018, Part C. A student performs a test in the morning and can recall the answers in the afternoon. Which of the following is most likely involved in the consolidation of memory during the day? A. NMDA receptor activation. B. GABA receptor upregulation. C. CREB inhibition. D. Inhibition of calcium influx. Answer A. Explanation. NMDA receptor activation is essential for long-term potentiation and memory consolidation. CSIR Net Deck 2015, Part C. Blocking protein synthesis shortly after learning a task effects. A. Only short-term memory. B. Only long-term memory. C. Both short and long-term memory. D. No effect on memory formation. Answer B. Explanation. Long-term memory requires new protein synthesis, unlike short-term memory. CSIR Net June 2021, Part B. The hippocampus is primarily involved in which type of memory? A. Procedural memory. B. Classical conditioning. C. Declarative memory. D. Priming memory. Answer C. Explanation. Declarative memory, episodic and semantic, relies on the hippocampus for encoding and consolidation. Would you like the next lecture on biological clocks, sleep and memory, learning and memory integration, or neural circuits and behavior? Let me know, and I'll prepare it just as thoroughly. Memory, complete theory for CSIR Net JRF Life Sciences. Memory is the fundamental process by which organisms encode, store, and retrieve information. It is a vital function of the nervous system that allows an individual to adapt, learn from past experiences, and make decisions. At its core, memory is a consequence of neuroplasticity, whereby synapses, neuronal networks, and entire brain structures undergo biochemical, electrical, and morphological changes in response to experience. Memory can be categorized into various types based on time scale, consciousness, and neural circuits involved, including sensory memory, short-term memory, working memory, and long-term memory, which can be further divided into explicit declarative and implicit non-declarative forms. Sensory memory is the most immediate form of memory, lasting less than a second. It temporarily retains sensory impressions, like visual, iconic, and auditory, echoic information, and serves as the initial filter before the information is processed further. Sensory memory depends largely on activity in the primary sensory cortices. Short-term memory, STM, retains limited information for a brief duration, usually seconds to minutes. The classic example is remembering a phone number long enough to dial it. Working memory is a refined component of STM that not only stores but also manipulates information in real time, such as solving a math problem mentally. Working memory is highly dependent on the prefrontal cortex, where executive control, attention regulation, and integration of multiple cognitive processes occur. Long-term memory, LTM, refers to the enduring storage of information for days, years, or a lifetime. It involves two major subtypes: explicit memory, which requires conscious recall, and implicit memory, which operates unconsciously. Explicit memory includes episodic memory, personal experiences, and semantic memory, facts and knowledge. These types are primarily associated with the hippocampus, medial temporal lobe, and neocortex. The hippocampus is critical for the consolidation of short-term episodic memories into long-term storage. Lesions in this area, as seen in the famous case of Henry Molaison, HM, result in profound anterograde amnesia, loss of the ability to form new long-term memories. Implicit memory includes skills and habits, procedural memory, classical conditioning, and priming. It relies on brain areas like the basal ganglia, cerebellum, and amygdala. Procedural memory enables learning of motor sequences, e.g., riding a bicycle, and does not require conscious recall. At the cellular level, memory formation involves changes in synaptic strength via processes like long-term potentiation, LTP, and long-term depression, LTD, particularly in the hippocampus. LTP, triggered by high-frequency stimulation, leads to increased calcium influx through NMDA receptors, activation of CA-McKee and PKC, and subsequent insertion of AMPA receptors into the postsynaptic membrane, resulting in stronger synaptic responses. Over time, LTP induces gene expression changes in protein synthesis necessary for stabilizing synaptic modifications, a process called memory consolidation. Transcription factors like CREB, GAMP response element binding protein, are essential for initiating these gene expression cascades. Blocking protein synthesis immediately after learning impairs long-term memory but not short-term memory, reinforcing the molecular distinction between memory stages. In addition to the hippocampus, sleep plays a major role in memory consolidation, especially for declarative memories. During sleep, hippocampus-dependent memories are repeatedly reactivated and redistributed to cortical sites for long-term storage. This supports the two-stage model of memory, where hippocampal networks initially encode information, which is later transferred to the neocortex for permanent retention. Forgetting, often misunderstood as memory failure, can be an active process, necessary to prevent cognitive overload and optimize decision-making. Forgetting involves mechanisms like synaptic pruning, limited, and inhibitory regulation of memory retrieval circuits. The prefrontal cortex modulates the recall and suppression of memories, balancing memory retention with adaptability. Neurodegenerative diseases, such as Alzheimer's disease, are characterized by severe memory impairments. Alzheimer's typically begins with loss of episodic memory, progressing to semantic and procedural deficits. The pathological hallmarks include amyloid beta plaques, neurofibrillary tangles, and hippocampal atrophy, all of which disrupt the neural circuits essential for memory. In summary, memory is a multi-layered process involving encoding, storage, and retrieval of information through complex and interdependent neural, cellular, and molecular mechanisms. From synaptic plasticity to large-scale neural networks, memory illustrates how behavior, experience, and biology are intimately connected. Its neural basis is not only central to neuroscience but also critical for addressing cognitive disorders, optimizing learning strategies, and developing therapeutic interventions. Previous year CSIR Net questions on memory, CSIR Net June 2018, Part C. A student performs a test in the morning and can recall the answers in the afternoon. Which of the following is most likely involved in the consolidation of memory during the day? A. NMDA receptor activation. Receptor upregulation. CCR/EB inhibition. D. Inhibition of calcium influx. Answer A. Explanation. NMDA receptor activation is essential for long-term potentiation and memory consolidation. CSIR Net Deck 2015, Part C. Blocking protein synthesis shortly after learning a task effects, only short-term memory. Only long-term memory. C. Both short and long-term memory. D. No effect on memory formation. Answer B. Explanation. Long-term memory requires new protein synthesis, unlike short-term memory. CSIR Net June 2021, Part B. The hippocampus is primarily involved in which type of memory? A. Procedural memory. B. Classical conditioning. C. Declarative memory. D. Priming memory. Answer C. Explanation. Declarative memory, episodic and semantic, relies on the hippocampus for encoding and consolidation. Would you like the next lecture on biological clocks, sleep and memory, learning and memory integration, or neural circuits and behavior? Let me know, and I'll prepare it just as thoroughly. You said, cognition, ChatGPT said. Here is your CSIR Net JRF Life Sciences full-length lecture on cognition, written at maximum detail, entirely in paragraph format, with no tables, and tailored to the JRF level based on previous year question trends. Includes 2 to 3 solved previous CSIR Net questions with year and answers, and is optimized to serve as your exclusive, all-in-one coaching material for exam success. Cognition, complete CSIR Net JRF Life Sciences lecture. Cognition refers to the mental processes and faculties involved in acquiring, processing, storing, and using information. It encompasses a wide array of high-level brain functions, including perception, attention, learning, memory, language, reasoning, problem-solving, and decision-making. In biological terms, cognition is not simply abstract thought, it is deeply rooted in neural architecture and shaped by evolution, experience, and environment. The cognitive capabilities of an organism directly impact its survival, reproduction, and social dynamics, making cognition a central theme in behavioral neuroscience, evolutionary biology, and psychology. At the neural level, cognition arises from coordinated activity within and across different regions of the central nervous system, particularly the cerebral cortex, which houses areas such as the prefrontal cortex, temporal lobe, and parietal cortex, all of which play specific roles in cognitive operations. Prefrontal cortex (PFC) is the executive hub of cognition, responsible for attention regulation, planning, impulse control, working memory, decision-making, and social behavior. Lesions or dysfunctions in the PFC often result in cognitive deficits such as poor judgment, impulsivity, or inability to plan future actions, as seen in frontal lobe syndrome and various psychiatric disorders. Perception is the first cognitive step, involving the interpretation of sensory input from the environment. The primary sensory cortices receive inputs e.g., visual, auditory, tactile, which are then integrated in association areas to generate a meaningful representation of the environment. From this base, cognitive functions such as attention selectively enhance relevant stimuli while suppressing distractions, a process mediated by dopaminergic and noradrenergic neuromodulation in the PFC and parietal cortex. Learning and memory, as already discussed, serve as cognitive foundations for the storage and retrieval of past experiences. Working memory, the transient holding of information for manipulation, is a crucial component of cognition and is tightly regulated by prefrontal circuits in coordination with parietal and hippocampal regions. Importantly, cognitive flexibility, or the ability to adapt thinking and behavior in response to changing environments or roles, depends on the dynamic interplay between the prefrontal cortex and subcortical structures such as the basal ganglia. Language, one of the most sophisticated cognitive functions, involves specialized brain regions in humans, Broca's area for speech production and Wernicke's area for language comprehension. While complex language is unique to humans, other animals exhibit forms of symbolic communication and vocal learning, offering comparative insight into the evolution of cognitive processes. In birds, for example, song learning in zebra finches requires dedicated neural circuits like the high vocal center (HVC) and RA nucleus, which are shaped by both genetics and experience. Decision-making and problem-solving represent the most advanced levels of cognition. They require evaluating alternatives, anticipating outcomes, and choosing optimal strategies. These are influenced by both rational computation and emotional modulation, the latter involving structures such as the amygdala and orbitofrontal cortex. Neuroeconomic studies reveal that even non-human primates and rodents exhibit rudimentary forms of decision-making, including risk assessment and delayed gratification, illustrating that the roots of cognitive processes extend deep into the animal kingdom. Social cognition, the capacity to perceive, interpret, and respond to the behaviors and intentions of others, is crucial in animals that live in complex societies. It involves theory of mind, empathy, and recognition of social hierarchies. Primates, elephants, dolphins, and certain birds such as corvids show impressive cognitive abilities such as mirror self-recognition, imitation, and tool use. These behaviors are interpreted as evidence of higher-order cognition, supported by evolutionarily expanded cortical regions and enriched life experiences. On a molecular and cellular level, cognition is underpinned by synaptic plasticity, neurotransmitter dynamics, and gene regulation. Glutamatergic signaling, especially via NMDA receptors, is central to synaptic changes involved in learning and problem-solving. Modulators like dopamine and serotonin influence motivation, reward-based learning, and mood-dependent decision-making, which are integral to cognitive engagement and performance. Cognitive deficits, therefore, often result from imbalances or dysfunctions in these systems, as seen in disorders like schizophrenia, ADHD, autism, and Alzheimer's disease. Importantly, cognition is not static, it is plastic and modifiable through environmental enrichment, experience, and training. Studies on neurogenesis in the adult hippocampus suggest that learning and cognitive challenges promote neuronal birth and integration, reinforcing the adaptive nature of cognition. This neuroplasticity forms the basis of cognitive therapies and rehabilitation strategies in clinical neuroscience. In summary, cognition integrates sensory perception, memory, emotion, attention, and decision-making through a network of brain regions, neural circuits, and molecular pathways. It is dynamic, context-dependent, and a product of both evolution and learning. For CSIR Net Sciences, understanding cognition provides crucial insight into how organisms perceive and interact with their world, a key theme linking neurobiology, behavior, and evolution. Previous year CSIR-NET questions on cognition, CSIR-NET Deck 2014, Part C. Which brain region is primarily associated with higher cognitive functions such as decision-making, planning, and attention regulation? A. Hippocampus. B. Amygdala. C. Prefrontal cortex. D. Cerebellum. Answer, C. Explanation. The prefrontal cortex is the seat of executive functions critical to cognition. CSIR-NET June 2019, Part C. Which of the following statements about cognition in animals is true? A. Only primates show tool use in planning behavior. B. Cognition is exclusively dependent on language. C. Certain bird species exhibit cognitive traits like problem-solving and tool use. D. Cognition cannot evolve in non-mammalian species. Answer, C. Explanation. Birds like crows and parrots demonstrate problem-solving and tool use, indicating advanced cognition. CSIR-NET Deck 2020, Part B. The neurotransmitter involved in modulating reward-based decision-making and cognitive tasks is GABA. B. Dopamine. C. Serotonin. D. Acetylcholine. Answer, B. Explanation. Dopamine is crucial for reward prediction, motivation, and reinforcement learning. Let me know if you'd like the next lecture on biological clocks, sleep, emotion, learning models, or any other CSIR-NET topic. I'll prepare it with the same quality and detail. You said, sleep and arousal, ChatGPT said. Here is your JRF Life Sciences full-length lecture on sleep and arousal, written with maximum detail, no tables, and tailored to the JRF level based on previous year question trends. It includes 2 to 3 solved previous year CSIR-NET questions with year and answers, and is designed to serve as your exclusive and final coaching material to help you crack the exam. Sleep and arousal, CSIR-NET JRF Life Sciences lecture. Sleep is a highly conserved physiological process observed across the animal kingdom, characterized by a reversible state of reduced responsiveness to external stimuli, decreased movement, and specific neurophysiological patterns. It is essential for survival, optimal brain function, memory consolidation, metabolic regulation, and synaptic homeostasis. The process of sleep is regulated by intricate interactions between circadian rhythms, homeostatic drive, and neurotransmitter-mediated arousal systems. Sleep is broadly classified into 2 main stages: non-rapid eye movement (NREM) sleep and rapid eye movement (REM) sleep. NREM sleep consists of multiple stages, progressing from light to deep sleep, marked by progressive synchronization of electroencephalogram (EEG) activity. In contrast, REM sleep is characterized by desynchronized EEG patterns resembling wakefulness, rapid eye movements, and muscle atonia. REM sleep is most closely associated with vivid dreaming and is crucial for emotional regulation and memory integration. The regulation of sleep involves complex neural anatomical circuits. The hypothalamus, specifically the ventrolateral preoptic nucleus (VLPO), plays a critical role in sleep initiation by inhibiting arousal-promoting centers through the release of inhibitory neurotransmitters such as GABA and galanin. The brainstem and basal form contain wake-promoting centers such as the locus ceruleus, noradrenaline, raphe nuclei, serotonin, tuberomammillary nucleus, histamine, and cholinergic neurons, all of which interact with the thalamus and cortex to sustain arousal and attention. Arousal, in contrast, refers to the state of alertness or wakefulness, allowing organisms to respond actively to external stimuli. The reticular activating system (RAS) in the brainstem is pivotal for maintaining cortical activation and attention. Arousal levels can range from drowsiness to full alertness and are modulated by sensory inputs, emotional states, and circadian influences. The ascending arousal system is composed of multiple neurotransmitters, including dopamine, acetylcholine, norepinephrine, and orexin hypocretin, each contributing to different dimensions of alertness and cognitive engagement. One of the most crucial regulatory mechanisms in sleep-wake regulation is the circadian clock, located in the suprachiasmatic nucleus (SCN) of the hypothalamus. The SCN receives direct input from intrinsically photosensitive retinal ganglion cells (IPRGCs), allowing light to synchronize endogenous rhythms with environmental day-night cycles. Circadian regulation ensures the temporal alignment of sleep with the dark phase in diurnal animals. Disruption in this alignment, as seen in shift workers or jet lag, can impair sleep quality and lead to cognitive dysfunction, metabolic disturbances, and mood disorders. Homeostatic sleep drive is another major component, governed by the accumulation of sleep-promoting substances such as adenosine during wakefulness. Adenosine is on A1 receptors in the basal forebrain to inhibit arousal-promoting neurons, facilitating the transition to sleep. Caffeine, a common stimulant, exerts its wake-promoting effects by blocking adenosine receptors. Sleep has profound effects on learning, memory, immune function, and metabolism. Slow-wave sleep (SWS) during NREM is essential for consolidating declarative memories, while REM sleep supports procedural memory, creativity, and emotional processing. Chronic sleep deprivation impairs cognitive performance, reduces alertness, and disrupts hormonal balance. In experimental studies, animals deprived of REM sleep showed increased irritability, anxiety-like behaviors, and even death after prolonged deprivation, emphasizing the essential nature of sleep. The flip-flop switch model describes how sleep and arousal states are stably maintained. In this model, VLPO neurons inhibit arousal centers and vice versa. Mutual inhibition between sleep-promoting and wake-promoting neurons ensures a rapid and stable transition between sleep and wake states. However, this system can become unstable in conditions such as narcolepsy, a neurological disorder characterized by excessive daytime sleepiness and sudden transitions into REM sleep. Narcolepsy is associated with a deficiency of orexin hypocretin, a neuropeptide produced by neurons in the lateral hypothalamus that stabilizes the sleep-wake switch. Another condition, insomnia, involves the inability to initiate or maintain sleep, often due to hyperarousal. It is commonly associated with stress, anxiety, or disrupted circadian rhythms. In contrast, REM sleep behavior disorder involves the loss of muscle atonia during REM, leading to dream and acting behaviors. These disorders underscore the complex balance of neural mechanisms that sustain sleep and arousal. From an evolutionary perspective, sleep may have developed to reduce energy expenditure, avoid predators during vulnerable periods, and facilitate brain plasticity. Comparative studies in animals such as fruit flies, Drosophila zebrafish, and mammals demonstrate sleep-like states and reveal conserved genetic and molecular pathways, including period per and timeless TIMP genes involved in circadian rhythm regulation. In sum, sleep and arousal represent dynamic, interdependent states governed by neural circuits, neurotransmitters, and genetic regulators. Understanding these processes provides crucial insight into how organisms manage energy, process information, and maintain internal physiological balance. For CSIRNet Life Sciences, a deep understanding of these mechanisms is vital for tackling questions that intersect neurobiology, behavior, and physiology. Previous year CSIRNet questions, Sleep and Arousal CSIRNet Deck 2017, Part C. Which of the following brain regions is primarily involved in the regulation of sleep-wake cycles? A. Hippocampus. B. Suprachiasmatic nucleus, SCN, C. Amygdala. D. Cerebellum. Answer, B. Explanation: the SCN is a master circadian pacemaker coordinating daily sleep-wake rhythms. CSIRNet June 2014, Part B. Which neurotransmitter is most closely associated with promoting wakefulness and is deficient in narcolepsy? A. GABA. B. Dopamine. C. Orexin, hypocretin. D. Serotonin. Answer, C. Explanation: orexin stabilizes the sleep-wake transition, its loss causes narcolepsy. CSIRNet Deck 2016, Part C. REM sleep is characterized by which of the following features? A. Muscle rigidity and slow brain waves. B. Rapid eye movement, muscle atonia, and dream activity. C. Increased muscle tone and delta waves. D. Absence of EEG activity and eye movement. Answer, B. Explanation: REM sleep includes rapid eye movements, vivid dreams, and muscular atonia. Let me know if you'd like the next lecture on biological clocks, emotion, learning theories, or any other topic from the CSIRNet syllabus. I'll make it just as complete and exam-oriented. You said, biological clocks. ChatGPT said. Here is your CSIRNet JRF Life Sciences full-length lecture on biological clocks, written in maximum detail, paragraph format with no tables, and tailored to the JRF level. It focuses on previously asked question areas and includes 2 to 3 CSIRNet previous year questions with year and answers. This is designed as your final, exclusive coaching material, helping you master this topic thoroughly for the exam. Biological clocks, CSIRNet Life Sciences lecture. Biological clocks are internal time-keeping mechanisms that allow organisms to regulate physiological, biochemical, and behavioral processes in synchrony with predictable environmental cycles such as the day-night rhythm, lunar cycle, tidal changes, or seasonal variations. These clocks enable organisms to anticipate changes in the environment rather than merely reacting to them, thereby conferring a significant evolutionary advantage. The most well-studied biological rhythm is the circadian rhythm, which follows an approximately 24-hour cycle. Circadian rhythms regulate daily fluctuations in sleep-wake cycles, hormone secretion, body temperature, feeding behavior, and gene expression. These rhythms are generated by endogenous oscillators, meaning they continue even in the absence of environmental cues, like light, although they can be synchronized or entrained by external time cues called Zeitgebers, German for time givers, the most potent of which is light. The suprachiasmatic nucleus, SCN, of the hypothalamus is the principal pacemaker in mammals. Located above the optic chiasm, the SCN contains a set of neurons that generate rhythmic patterns of activity via transcriptional translational feedback loop, TTFL. The neurons maintain rhythmic expression of clock genes, which include clock, BMAL1, PER period, and cryptochrome. Clock and BMAL1 heterodimerize and promote the transcription of BRN CRY. As BRN proteins accumulate in the cytoplasm, they form complexes that translocate to the nucleus and inhibit the activity of clock BMAL1, thereby repressing their own transcription. This creates approximately 24-hour oscillation in gene expression. In addition to clock in the SCN, peripheral clocks exist in other tissues such as the liver, lungs, kidney, and even individual cells. These peripheral clocks are also based on the same molecular feedback loop but are entrained by systemic signals such as feeding, body temperature, and hormones like glucocorticoids. The SCN synchronizes these peripheral clocks, ensuring organism-wide temporal coordination. Photoreception and light entrainment of the circadian clock involve a specialized set of retinal ganglion cells expressing melanopsin, which are intrinsically photosensitive and distinct from rods and cones. These cells project directly to the SCN via the retinohypothalamic tract, allowing light information to reset the circadian rhythm. This is particularly important for entraining the sleep-wake cycle to the external light-dark cycle. The output of the SCN regulates the pineal gland, which secretes the hormone melatonin during the dark phase of the light cycle. Melatonin feeds back to the SCN and other tissues, acting as a hormonal signal for darkness and helping regulate sleep timing, seasonal rhythms, and reproductive cycles in photoperiod-sensitive organisms. Ultradian rhythms are cycles shorter than 24 hours, e.g., the basic rest activity cycle or hormone secretion cycles, whereas infraradian rhythms exceed 24 hours, e.g., menstrual cycle or seasonal breeding. All these biological rhythms can be governed by similar oscillator mechanisms, although the circadian clock is the best characterized. In model organisms like Drosophila melanogaster, the genetic and molecular basis of biological clocks has been extensively dissected. In Drosophila, PER and TIM are the core feedback repressors, and their expression oscillates over the course of the day. Mutations in period PER and timeless STIMP genes cause changes in the free-running period or even abolish rhythmicity altogether. These discoveries led to awarding of the 2017 Nobel Prize in Physiology or Medicine for the elucidation of molecular mechanisms controlling circadian rhythms. Disruptions in biological clocks can have profound health consequences. Circadian misalignment, such as that seen in shift workers or individuals with jet lag, leads to impaired cognition, metabolic dysfunction, depression, and increased risk of chronic diseases. Circadian clock also affects drug metabolism, and thus the field of chronopharmacology explores how the timing of drug administration can influence therapeutic efficacy and toxicity. Studies of evidence in humans have shown that lesions to the SCN abolish circadian rhythms of activity, hormone secretion, and body temperature. Transplantation of SCN tissue can restore rhythmicity, demonstrating the autonomy and hierarchical control of the central clock. Even in plants and cyanobacteria, circadian rhythms are generated by transcriptional or post-translational feedback mechanisms. In cyanobacteria, the Chi-ABC system produces a self-sustained rhythm and phosphorylation status of proteins, independent of gene transcription. From an evolutionary perspective, biological clocks enhance fitness by optimizing the timing of key activities such as feeding, reproduction, and predator avoidance. For example, nocturnal and diurnal species exhibit contrasting circadian phase relationships with respect to melatonin secretion and core body temperature rhythms, illustrating the adaptability of internal clocks to ecological niches. Biological clocks are ubiquitous and tightly regulated systems that orchestrate the timing of physiological and behavioral processes. Understanding their molecular basis, neural circuitry, and physiological outputs is vital for decoding how organisms maintain homeostasis in a rhythmic environment. For CSIRnet Life Sciences, mastering this topic means not only grasping the molecular feedback loops and brain regions involved but also their ecological, physiological, and pathological implications. Previous year CSIRnet questions, Biological Clocks, CSIRnet Deck 2015, Part B. Which of the following brain regions acts as the master circadian pacemaker in mammals? A. Hippocampus. B. Suprachiasmatic nucleus, SCN, C. Amygdala. D. Prefrontal cortex. Answer, B. Explanation: the SCN in the hypothalamus orchestrates circadian rhythms across the body. CSIRnet June 2016, Part C. Mutations in the PER and TIM genes in Drosophila disrupt which biological process? A. Wing development. B. Reproductive behavior. C. Circadian rhythm regulation. D. Chemosensory signaling. Answer, C. Explanation: the PER and TIM genes encode core components of the fly circadian clock. CSIRnet Deck 2017, Part B. Which hormone, secreted in darkness, is regulated by the SCN and involved in circadian rhythm modulation? A. Cortisol. B. Insulin. C. Melatonin. D. Ghrelin. Answer, C.